I thank the organizer for inviting me to speak at this important conference. I'm Matteo Becatti, Associate Professor of Biochemistry at the University of Florence, and I'm glad to introduce you a novel device for measuring thrombotic risk. Thrombosis is responsible for 1 in 4 deaths globally, and occurs when a thrombus, blood clots, form inside the blood vessel, obstructing normal blood flow. Thrombosis can lead to ischemia and conditions like stroke, myocardial infarction, or pulmonary embolism. Early diagnosis is crucial. Current methods fall short in predicting thrombotic events, and we need for more precise tool that accurately assess thrombotic risk and prevents related events. Our previous data demonstrate the key role of fibrin structure, of the three-dimensional structure of the clot in promoting thrombosis. Clot permeability reflects the pore size in the fibrin network and is a critical factor in understanding clot structure. Reduced clot permeability is linked to recurrent thrombotic events in stroke, pulmonary embolism, or deep vein thrombosis patients. As you can see here, this is an in vitro clot from control and from high cardiovascular risk patient. What is the difference? The difference is permeability. The clot derived from controls is more permeable and bigger for respect to control from high cardiovascular risk patients. Current methods for measuring clot permeability are imprecise and lack standardization from clinical use. We need for a more reliable and repeatable system for assessing individual thrombotic risk. For this reason, we've proposed a new portable, cost-effective device designed for use in clinical setting. This device measures clot permeability using a pressure-based system based on Darcy law. Our device is designed to apply consistent overpressure to the liquid surface and measure droplet weight over time from the clot. It uses a STM32 microcontroller for sensor data acquisition, overpressure regulation, and PC communication. And it includes a LabVIEW-based interface for data acquisition and analysis. Here we can see in detail the structure of our device, the air pump that pushes water here into the clot, insert here in this filter, the microcap, the water container, the microbalance, the pressure sensor, and the microcontroller. Our preliminary experiment using plasma and purified fibrinogen from 30 controls and 30 high cardiovascular risk patients demonstrate that our device is able to distinguish control from high cardiovascular risk patients. In conclusion, to assess thrombotic risk, a low-cost automated system for measuring clot permeability has been developed in collaboration with engineers from University of Siena and clinicians from University of Firenze. Preliminary data show that the proposed device is rapid, precise, and requires minimal samples, making it suitable for clinical use. Obviously, more studies with bigger sample size are required to confirm this data. Thank a lot Professor Valerio Vignoli, Professor Ada Fort, and Professor Marco Mugnaini from University of Siena, the engineers group, and Professor Rossella Marcucci, Professor Domenico Prisco, and Professor Giacomo Emi from University of Florence, clinicians, and also my lab. Thank you very much.